Hey guys, Steve here, Aquaponics. Today we're going to talk about
Growing with fishes. Growing with fishes.
And data. I know at least once a month I'm sending him some pictures or trading pictures or documenting something I think he might find interesting and sending it off to him. So definitely excited to have him on. Alrighty, let's pick Super Lemonade. We're gonna pick you because we're about to launch a lemonade in Oklahoma. Super Lemonade. If you want to email me at potentponics@gmail.com, we will get you your silicate. Thanks a lot. All right, Matthew, take it away.
Get us one more for potassium silicate.
What's up?
Get us one more person for potassium silicate.
We'll do it on the next break.
All right, that'll work.
All right. Let me, I think he's muted there.
I see. Can you hear me now?
There we go. Now we can hear you.
Yeah, well, I appreciate the compliment. Thank you very much. I think I'll have to share my screen, right? Is that not the case? Hello?
Yep, you should be able to. Let me know if you have a problem there. We'll get you straightened out. There we go.
There we go. All right, you can all see this?
Yep.
Cool. So my name's Mathew Gates. I am an integrated pest management specialist, and I have been for the past 10 years, actually. What a year to end the decade on, huh? Well, I will agree with my friend here that he does often have really great pictures to send me. And for that reason, he inspired me to talk about one of the pests that I will be going over, which are just moth larvae in general. Anyways, here I'm going to talk about some sort of basic and advanced aspects of integrated pest management, and also bring in some aspects that are relevant to aquaponics in particular. And then also we're going to talk about some of the pests that I see both in aquaponics areas and just kind of generally, especially this time of the season. So it's a little bit of a seasonally inclined iteration of this presentation. You can find me on Instagram and Twitter at SyncAngel, and you can also find me as Zenthanol on YouTube. And I have a bunch of integrated pest management videos about various pests and evolution and cannabis ecology and its origins and all this sort of thing. So if you're interested in that information, you can check out my YouTube channel. But here's the bottom line up front. So I like to characterize the different kinds of integrated pest management sort of techniques or actions that you can make in sort of like groups. Usually I put them into 5 groups that I normally name, and sometimes I'll change them a little bit, and I'll get into that. But essentially they're the biological, the chemical, the cultural, the genetic, and the mechanical types of controls. Biological controls are kind of self-explanatory. They are living agents. Generally, I also like to put viruses into this category as well, although there aren't a whole lot of viruses that are used in integrated pest management, but that might be changing in the future. I know that's a scary subject right now, but there are some beneficial viruses, plant viruses in particular, and mycoviruses that infect pathogens of plants that are fungi. And so there is some possibility for using them as sort of a natural biocontrol agent, I suppose. although some definitions of life don't include them. There's also chemical controls. Those can be synthetic or they can be naturally occurring. Of course, many synthetic pesticides that are used in agriculture cannot be used in cannabis or should not be used in cannabis. A lot of times that's because they are systemic. And it's important to know that there are different kinds of pesticides. Some of them are systemic, so that means that when they get into the plant, they actually move through the tissues. They can move up and down to various degrees. It depends on the compound. And that's the reason why people don't like to use them in cannabis. There's also regulations against this, of course. There's also translaminar chemical pesticides. These are pesticides that do get into the tissue and don't stay just outside of the tissue, but they don't actually travel throughout the plant like a systemic pesticide would. And then you have pesticides that don't have either of those sort of effects, and they simply stay as residue on the plant. Maybe they last a long time, maybe they last a very short time, maybe they're very photoreactive and so they decay in sunlight or ultraviolet radiation or something like this. You've also got cultural controls. Cultural controls are sort of like your procedural controls. They're how you go about doing things, your SOPs and that sort of a thing. And whether you're growing on a small scale or a commercial scale, this is an extremely important aspect because in my opinion, most of the controls that people like use or actually use to affect change within their crop are these cultural controls, and they tend to be very cost effective. They might be something as simple as changing how you harvest or changing one of the products you use to, or whatever process you use to like cure the final product. Or in the case of cultivation, maybe it's how you prune your plant, or maybe it's how you train it or something like this. Or maybe it's the fact that you have caulked up a bunch of holes in your facility or your garage or wherever you're growing so that pests can't get in or something like this. There are also genetic controls I like to call. Usually you don't have very much effect on this once you're actually growing plants and crop. This is more of a thing that is affected by your pedigree, your breeding, the genotype of the plant sort of, and its resistances and susceptibilities on a genetic level. For example, hemp, so in some cases, plants that don't have the ability to produce THC, cannabis plants in particular, some of them have a gene cassette, a sort of a string of DNA that, or genes rather, that are that have the THC synthase genes that are important for that pathway to create that cannabinoid, that metabolite. But they also lack certain resistance genes to fungi. And a company called Medicinal Genomics has been looking into this and has published information and research on this. And so it turns out that plants that lack this gene cassette are naturally more susceptible to certain fungal pathogens. So it's just a thing to consider. This is true for other kinds of crops as well. Basically, all kinds of plants have genetic susceptibilities and weaknesses that pests and other sorts of environmental conditions can have a negative or positive effect on. And then finally, we have mechanical controls. Those are things that are going to be based on physical destruction of the organism. Although a lot of people are used to thinking of like chemical like interactions and that sort of a thing, things like ultraviolet radiation cause physical damage by like disrupting the genetic makeup. It's one of the reasons why sunburns can be so bad. It's one of the reasons why skin cancer happens in the way that it does. It's why melanin is so helpful for availing yourself of that and getting rid of that problem. A lot of insects you might have noticed and mites for that matter, will feed on the undersides of leaves. And there's also a predator sort of evasion behavioral reason for this, but also it's because of the UV radiation. A lot of organisms are sensitive to UV radiation in some way. It's one of the first things we had to sort of adapt to when we came out of the ocean, so to speak. There are other mechanical controls like literally just squishing and killing a pest, right? The I in IPM is your integration. The key is to be able to integrate all of these different factors together so that you put as many advantages on your side and levy as many disadvantages to the pest side, whether they're an arthropod like an insect or a mite, or whether or not they're a fungal pathogen or a bacterial pathogen. You have to you have to consider all of the possibilities when it comes to getting rid of pests and preventing them, both in a curative way, but also in a prophylactic way. In this example, I have biological control. We have a predatory mite, Embasius swirskyii. We also have a yellow sticky card as a mechanical trap, which generally these small ones are primarily used to tell what kind of pests you're dealing with. They're not actually meant to be very good in the way of actual control. But this is sort of an example. You can get sticky cards and rollers that are larger and kind of sticky tape that you can apply as well. There's also mesh screens and barriers, which are another kind of mechanical control because they don't cause any lethal damage, but they prevent things like moths and large insects, and even small insects in the case of thrips screen, which I am a big advocate for as a mechanical barrier to keep things from coming in. That gets rid of most of your problems on the onset. You also have crop scouting, which is incredibly important, and I would call that a cultural control. It's a process or standard by which you go about doing things, and it's important, and I train people on this, how they go about it, how they take samples, what are they looking for, both in the case of pests or their damage, or abiotic factors like wilting or problems with like the piping in your facility or that sort of thing. There are non-biological aspects of crop scouting as well to consider, but mainly from the biological side. You are tracking whether the beneficials are being effective as well if you're using any. You're checking for signs of like parasitation or predation or that sort of thing, and just generally if you see those organisms after you release. If you don't and you're using certain chemicals or maybe you're applying something else, you might consider, well, maybe I'm negatively affecting my biological controls. And then you can just simply remove pests as well, which I would also call a mechanical. In this iteration, I have a mechanical heavy IPM sort of example here. But there are many other ways to go about IPM, and every context is different. I did already give an example of different biological controls. I have 2 videos down here. I'll probably not play them, but there are 2 biocontrol examples here. On the left, we have Cryptolaemus montroseri, which is the mealybug destroyer. It's a lady beetle that actually feeds on mealybugs, not on aphids, for example. In fact, the lady beetle group is very varied. Some of them are even herbivores. Other ones feed on fungi, but many of them feed on insects in particular. You've also got your enzyme pathogens. So that includes like Beauveria bassiana, or Bacillus subtilis, or Bacillus thuringiensis, and its different isolates. So biocontrols can be microarthropods. They can also be larger things like birds and lizards that might exist in your natural area. But they also don't necessarily, they're not necessarily as controllable or commercially available. Moving on to chemical controls, I'm giving here examples of different classes by how the pesticide or what kinds of things the pesticide affects. But pesticides are often categorized in chemical classes. So whether or not the chemical agent utilizes, or whether the chemical agent is a kind of structure or comes from a certain place. Yeah. Let me check the chat real quick. No, it's not. Oops, never mind. So we have like fungicidal agents like myclophenol, which of course you should not use in cannabis, but I bring it up here so that people know exactly what, here's what the name is. If anyone calls it myclo or refers to it like that, you actually know what I'm talking about or know what this is exactly. And that's a systemic fungicidal agent that you shouldn't be using, but many people have used for powdery mildews and fungi and that kind of thing. I mean, it works, but also it gets into your plant systemically, and that's not good for you.
I've also seen people test hot for it if they just happen to be directly next to an orchard from overspray. One thing I wanted to add on that.
That's a really good thing to add, actually, is that you might be doing everything right. This is also true for other pests, for that matter, for both the fungus you're going to get and also for other pests and biocontrols, for that matter. They don't care about your borders. There is spray drift. There is a great example where a few years ago, with the sort of dicamba ready, this herbicide ready soybean lot, there were people who were applying dicamba to them and they were fine, but because it was a really hot day, the pesticide volatilized and drifted across. So they didn't even have wind drift in the beginning, but because of the heat and the wind pickup, the compound volatilized, moved through a bunch of soybeans that weren't herbicide ready and just destroyed them all, defoliated all of them. It was like a large acreage, a large amount of damage and acreage damage. So it does happen. There's also things like silver nanoparticles, for that matter, or sulfur, which is a totally natural element. Usually in the wettable sulfur applications people use, you shouldn't be using that in flour, but you can use it for various situations in veg or kind of around your area. It just sort of depends on what you're dealing with, but it is a fungicide. It's also insecticidal and mitocidal, for that matter. And if you apply too much, it's also herbicidal. But the dose makes the poison. There are insecticidal agents like cyantraniliprole, which is another one of those sort of systemic insecticides you shouldn't be using. There's a product out that's kind of new called the Spear series, and it's actually, I guess you could call it a biopesticide too, but it's synthesized Blue Mountains funnel web spider venom, which is pretty fascinating to me. And so they synthesized it, but they cleaved off the parts that affect mammals and only kept the parts that affect arthropods. And I think the active ingredient of the Spear series is this omega kappa venom peptide, which is just really, really cool to me. So I thought I would add that as well, that these sort of new chemical agents, these new biological agents are coming out that I think will really help with the sort of truncating of our uses of noxious chemical agents. There's also mitocidal agents like bifenazate or hexythiazox or clofentazine, none of which you should be using in cannabis, but there's a lot of very common miticides that are out there that people are familiar with in the agricultural sphere, or they become familiar with some research and they somehow acquire them and utilize them. So you just got to be really aware of this sort of a thing. And it happens more often than you think. And then we have finally herbicidal agents like glyphosate or sodium chloride, regular table salt. Both of these things, people will probably already know this, will definitely affect your plants negatively. I've already mentioned some cultural controls earlier, but to go over some, we have, to put a name to it, cyclometrics, which is sort of that interaction between humidity and temperature. So when people talk about like vapor pressure deficit and that sort of a thing, this is kind of what they're talking about to a degree. And evapotranspiration, when the water goes into the soil, goes up the plant, and releases through the stomata, this physiological process, that definitely has interaction with the room. And if you don't really know what that is, you can be on the you can be behind the power curve with regards to certain fungal pathogens, for example, that are very sensitive to a change in temperature and humidity, because that initiates the germination of the spores in a lot of cases. It's what they use to be able to tell that the situation is ripe for germination and it's not too dry or too wet. Your labor operating procedure is a cultural control, which I kind of mentioned earlier, but how you go about things, all of this affects your IPM in minute ways and in very large ways, of course. Your physical construction of your setup is also incredibly integral to how you go about your IPM. Obviously, if you're growing in a tent or if you're growing in a like a small room in a residential area, those are going to have very specific differences. You might be doing multiple things at once even, or you're operating in a large facility or outdoors. This all matters. In addition to this, your biogeographic area matters as well. So where you are in the world is going to influence what pests you deal with and at what frequency, and year to year and season to season, what their changes in population will be as well. And then, of course, you have hygiene maintenance, how you keep your area clean. Do you use a lot of bioremediation efforts in an aquaponics setup? Of course, you have to keep things very hygienic, and you can't let things get into the water or allow things to foul. That's a very important aspect of aquaponics to consider, because, and also if you keep the temperature too high, I'm sure people have already mentioned this or might be already aware of this, but water pathogens can become a huge problem if the water temperature heightens. Do you have anything to say about that, Stephen?
Oh yeah, it's a big problem we see here in Oklahoma and in Texas and a lot of the southern aquaponic facilities. One of the best ways we found to mitigate that, and actually he's going to be on the panel tomorrow, someone who just installed one of these, Bain Howard of Vertica. Actually just installed a giant geothermal coil onto their main facility up in about 2.5 hours from where I'm at. What they do is they put these giant coils in the ground that circulates water through the system and then down underground, which bleeds it off into that 58 degree, 56 degree temperature and allows that heat to dissipate. And it's honestly the cost-effective way to do it with the water temperature, in terms of you don't have to have chillers, you don't have to have a ton of power, you can run it off of a solar panel and run it completely off grid, and it doesn't cost you anything to run. And then you can also hybridize that with, again, doing a similar method with air exchange and doing GAT systems, which I believe, again, Josh is talking about tomorrow.
That's excellent. You would have great insight. So let me continue. Genetic controls, I mentioned some genetic controls earlier, but to give you some specific names, we have RNAi, mediated crop protection, which is RNA interference. And so that's when you use, so plants actually have RNAi naturally. There are aspects of plant physiological processes that allow them to silence DNA. And that can be very useful when pathogens use sort of similar tactics to override or suppress the plant's immune system. And of course, there are possible avenues for utilizing this in an IPM perspective, but it's kind of a new sort of a discipline. So technologically, I don't think a lot of this will be out very soon, but hopefully sooner rather than later. Because it would be very targeted and you wouldn't be able to achieve this sort of targeted nature in this specific way when using other techniques. So there's a lot of advantage to that, and especially from an ecological or environmental perspective, if you can sort of use the natural RNAi sources, or if you can instill a particular kind of RNAi to a plant through breeding or some other process, you can achieve this sort of level of deft effect. You've also got general plant resistance. Now, I think it's really important to talk about the terminology of resistance. So a lot of people will have different definitions, and I don't think there's a lot of there are a few organizations that might define resistance or disciplines rather too, that will define these things differently. But for me, and I have a research report, or rather a recommendation from an organization that I can send to anyone who's curious about it. But resistance is, so you have three kinds of resistance. You have tolerance, which is when a plant can still grow relatively well, even though a pest is feeding on it, a particular species. And it's important to bring up that tolerance and resistance and all of these words, they are only going to be really effective if you give some sort of qualifier. It doesn't, you can't just say resistant, it's resistant. Resistant to what? And to that degree, is it resistant to all the different kinds of those things, or only specific species, or a specific isolate, for example? It's important to know, because plants have various ways of defeating different kinds of threats, and if you know that it's resistant, you should know what confers that resistance in the plant if possible. And that's something that I'm excited to see more and more in cannabis. But tolerance is when a plant can more or less grow pretty well, even with some level of the pest. Resistance is when the pest actually has problems, or it's difficult for the pest to actually establish on the plant, more or less. And then immunity is actually when the particular organism that we're talking about cannot colonize the plant. There's something very inherent about the plant that is just not possible for it to colonize. Maybe even extremely minimal colonization or something like this, but pretty much not at all. So there are some aspects of immunity where a plant is just simply not a suitable host, and that's like the most basic kind of immunity. It's just they're just too different from what the organism or pathogen or whatever is used to interacting with. Then we have gene drives, which are a method of enhancing the inheritance of a preferred trait. Through breeding, for example, you can establish this effect where it's essentially how resistance accrues in various pest populations through natural selection. Mechanical controls, I'll go over this pretty briefly. We've got trapping, which is pretty self-explanatory. Putting up sticky cards or putting up some sort of an adhesive, or possibly even using light traps or pheromone traps in order to kill the organism. Although a pheromone trap would kind of be like a chemical control and a mechanical control, right? You can straight up remove the pest, which is oftentimes really helpful, especially if you notice that you only have one plant that's kind of colonized by like spider mites, or if you have a really pernicious pest that's very serious, like maybe hemp russet mite, for example, it might be just the more strategically optimal choice to just bag that plant and destroy it, because, and then maybe apply some sort of a preventative or prophylactic biocontrol agent or application spray or something like this, because if the infestation gets to be too much, you might very quickly end up on the wrong side of the cost benefit analysis with your crop. You can use non-chemical sterilants. I already kind of mentioned UV radiation does this, but boiling hot temperatures does this, desiccation does this. There are various ways that are not chemical that you can sterilize an area, and I'm excited to see better ways of people being able to do that in residential sectors as well as in commercial sectors. You've also got various ways of hand killing, which can still be kind of useful, especially on a small scale, but it's a lot less effective, of course, at large scale. And then I have a very, very basic graphic here from the Crop Protection Research Institute that showed that integrated pest management, at least in vegetable crops, is really great. You get about $19 for every $1 that's spent on it. And as far as cannabis is concerned, I would say that you probably get quite a bit more money per dollar of investment. So it's very important that you prevent and sort of become forewarned, because to be forewarned is to be forearmed. And if you have the sort of resources that are going to allow you to achieve that objective, like some of the videos I have on my YouTube channel or on Instagram, I'm very happy to help people with insect identification or various other sorts of tasks that are important to get you started. It's very important to me that people are able to get access to cannabis and also good food and that sort of a thing. And if you can grow it yourself, or if you're getting it from somewhere else, you can at least assess and know how they're going about it. And then I have a few pests. Hopefully I'm good on time. Yeah. So, and after this, there's a Q&A, of course, right, Stephen? I think so.
Oh, yeah.
Yeah. Sorry about that. So the rice root aphids, the first one that I want to start on, of course, in an aquaponic setup where your roots are below, there are some, they're not a totally aquatic aphid. There's actually a sister species called the water lily aphid, and it is actually semi-aquatic. So the rice root aphid also has some of that trait. It can definitely do well in like a hydroponic setup. It can definitely colonize a cannabis plant and get at the stem and even parts of the roots that might be kind of out of the water somewhat, depending on how your setup is made, I suppose. They're considered to be originally from Asia and possibly, perhaps probably in the Japanese archipelago, and they have a different kind of a life cycle where they move from two different alternate host species. But in the West, in North America in particular, they don't tend to have this sort of normal life cycle, and that's probably because of not being in their native territory. A lot of aphids actually do this. There are two main groups of typical hosts. You have your grasses like wheat, sorghum, rice, obviously, rye, barley. And I find these kinds of plants often on people's properties growing kind of fairly. And if you're in a place where these grasses are grown commercially or residentially, then you should really be watching out for the rice root aphid, because many times I will do a site evaluation and I find that people dealing with a rice root aphid problem are the victims of the plants that they have on their property hosting the rice root aphid, especially your here stone fruits. You might have a plum tree that you never even realized, or people will say, oh yeah, this used to be a converted orchard, or this used to be this guy he used to grow various fruits on his property, and that's really great, right? And I say, yeah, it is really great, but also can be really great for pests. So you just have to be aware of this fact, and aphids in particular are really, really good about having multiple hosts if they're generalists anyways. And then just like all aphids, for the first two traits here, all aphids for the most part have telescoping generation, so they're born pregnant, and that makes them incredibly fecund. Maybe fecund isn't the right word, because it's usually clonal offspring. So they live birth usually. They don't lay eggs until it's the right season for them to do, and they produce a specific morph that has wings that can go out and do that, lay the eggs, overwinter, and then the nymphs can come out and recolonize the plants. Hopefully the plants were also successful, laid some seeds, and now the sprouts are coming out. That's one way that happens. You also have the production of honeydew. Aphids feed on the phloem sap channels of plants, and they've been doing this since before flowering plants ever existed. And they've gotten really good at taking a ton of sugar out of the phloem sap and using enzymes like sucrases and invertases and various other compounds that might be produced by their endosymbionts, which are bacteria that live in special cells. And they process all of the sugar and amino acids that are in the phloem. And they can't even do all of it at once, because the turgor pressure between the phloem sap and their bodies is so different that they're basically just tapping this really pressurized stream and just shunting it into their body. So they can't even get rid of all of it or process all of it at once physically. And that's why they produce honeydew, which is a super concentrated sort of sugary exudate that ants really like, and a lot of other organisms like honeydew as well. And you might find city molds that grow in the honeydew as sort of a black, molded looking thing. And it's not a direct pathogen to cannabis or other plants, but it does use honeydew as a substrate. So you have to watch out for that. It's also a great way to find out if you have aphids or another sort of hemipteran that might produce honeydew, like leaf hoppers or plant hoppers or that kind of a thing. And rice root aphids are somewhat water tolerant. I definitely see them in hydroponic setups especially well, and I have seen them in a couple of aquaponic setups as well, specifically deep water culture. Just some bionomic information. You can find this on my rice root aphid pest primer video on YouTube. But basically, they don't do very well at temperatures greater than or approaching 35 degrees Celsius. And I have here that a high temperature plus a RHPV infection could severely induce lifespan problems. And that's because we might have a potential ally for rice root aphid in the future, which is the Rhopalsiphum paddy virus. Rhopalsiphum paddy is another aphid species, is the bird cherry oat aphid. And this virus is known to infect many kinds of aphids, and it actually can exist in plants systemically. It doesn't cause problems for the plant, and it lasts in some plants anyways that's been tested in for about 2 weeks. And what's really crazy about this is that it doesn't outright kill the aphid, I suppose, but it does infect their ribosomes, and it heavily decreases their longevity and also their ability to propagate, which are two things that aphids really have going for them physiologically. So it's possible that this virus might be colonizable in other plants, and we might be able to take advantage of it in the future. We've also got Septoria Cannabis, which is a leaf spotting disease pathogen. There are actually very many Septoria out there, but this is one example of what the pathogen can look like on cannabis in particular. You see sort of like brown spotting. You might even see some hyphae growing out of the necrotic tissue that's sort of died before the brown spotting happens because of the dry circular infection points that it makes. Septoria Cannabis infects cannabis in specific, but I say that with a caveat because Septoria is one of those pathogens, one of those fungal pathogens that can hyperspecialize. And so it's kind of hard to say whether something's a species or a subspecies at that point, if you know what I mean.
I've seen 3 different distinct expressions of Septoria, and one thing I think I've noticed that I just wanted to add to this, when the early infection is, it often really looks like water spotting, like you've got water on it during a hot day. That's how almost always with all the different ones it starts to look like, and you go and you yell at the guy who was watering. Sorry about the beeping in the back, but you go and you yell at the guy who's watering, and then, the next day it's further up the plant and he didn't do it. And then you realize, oh, that's not the problem I had.
That's an excellent point to make, actually, because it's very true. It does look like water spotting. I've also had people misdiagnose this as, understandably, chemical burn from like a spray application or something, or if they're not used to seeing what that kind of looks like, they might be afraid that they've burned their plants or that's light burn or something like this. But the very circular nature of the infection is sort of a diagnostic characteristic that can help you achieve that level of identification. And I'll stress again that you might not necessarily know whether it's Septoria cannabis in particular, but this is one of the main species, and there's a reason why it's got the cannabis species epithet attached to it. But certainly other Septoria could infect cannabis, and like Stephen was saying. We've also got the dark-winged fungus gnats, which are part of the Berdesia genus. This is actually a frame from my pest primer on fungus gnats, in particular the dark-winged fungus gnats. This on the upper left is an example of what the fungus gnat looks like in adult. You might also be familiar with the larvae at the bottom there. They tend to, one way you can tell them from other kind of fly larvae is that they have a black head capsule, and also other worm-like things that you might find in your soil, like potworms or nematodes are very small as well. But fungus gnat larvae have this sort of characteristic cream-colored larvae or larval color. They've got the black head capsule, and despite their name fungus gnat, they don't necessarily just feed on fungus. In fact, they can feed on plant tissue just as easily as they can feed on fungal tissue. So don't think, because I've definitely run into people who've had this interpretation of the name, that they are specific to fungi, or that it means that you have fungal, that you have some sort of fungal population. You probably have some, but that you have a major one or a problematic level of fungi in your soil. That's not necessarily the case. Although it could very well become after the fact, because fungus gnats can vector various pathogens, and Botrytis cinerea is one of them, and it's definitely a fungus. And you can also get Fusarium solani. This is an example of Fusarium solani on the right that was vectored by fungus gnats in peas. I also have to tell you about the lettuce sclerosis virus. I've talked to Stephen a little bit about this too. But recently it was documented in the authorized farms in Israel. Currently it is incurable. Usually it often, like its name implies, infects lettuce, but also many other plants. And those plants are usually not worth, because they're annuals, sort of the process that we have to affect viruses, which is typically through tissue culturing and that sort of a thing. So usually if you get this pathogen, which is vectored by the silverleaf whitefly, Bemisia tabaci, which you can see in the upper right here, at that point, you kind of are at a loss. And that's kind of just the unfortunate reality at this point. It's very, very difficult to conventionally treat viruses. And although there might be some ways in the future that are much more economical and democratized, it's just not the case at the moment for the vast majority of cases, for a lot of pathogens for that matter, but viruses in particular. And that's due to the way that they systemically infect plants. One major problem with LCV in particular is that because it's vectored by this super vector of over 180 plus viruses, this silverleaf whitefly, it also, the host causes a yellowing when it feeds on leaves a lot of the time. Not all the time, but it can. So you don't really necessarily know, because the main symptom of leishmaniasis virus is chlorosis, that you're actually dealing with that chlorosis problem. It's only after much of the plant has become chlorotic that you might be more sure, but at that point, you probably haven't achieved control, and that would be a very sad thing to happen. And you can see some diagrams of cannabis in this chlorotic state. Now, just because you see chlorosis doesn't mean that you have the LCV, but there are places where you might be able to test. I believe Medicinal Genomics has a test for LCV as well as other cannabis viruses. I think also B. curlytop virus as well, which was also recently documented in the Midwest, particularly Colorado, and probably is in other places too, unreported.
I know Joshua Steenland documented it in Oregon last year.
That's right. I keep forgetting that point. I usually remember the Colorado example. Is there anywhere else that you can think of?
I've seen it this year in Oklahoma, or what I think it is, but I don't have testing for it, but it looks identical to what I saw in Oregon last year.
Yeah, you heard it here first, guys. Like that's a lot of different places, all Israel to, well, also I should say this, LCV is thought to originate in southwestern California. So that's another place. So if you're like me and you live in southwestern California, you should be aware of that. The Crini viruses in general are thought of as coming from this like southwestern North America area. Ironically, the silverleaf whitefly is considered to probably have originated in India based on a bunch of parasitoid wasps that also seem to specialize on whitefly. And if that's the case, those are two totally different biogeographic areas. So, it just kind of makes you wonder.
One other thing I wanted to add about the lettuce chlorosis virus is it's one big reason why you shouldn't be doing heavy lettuce production in the exact same system as your cannabis. You want to make sure you're running them in separate sump tanks, separate fish tanks. There's 2 aquaponic facilities that were DWC facilities that I've observed what we believe was lettuce chlorosis virus. This was before there was testing, but all the symptoms were there, and it sure as heck looked like it was on the lettuce. The lettuce stuff is heavily documented. So we were, again, on 2 separate grows, we were able to basically wipe everything out and then do a lactobacillus treatment and just let it sit for a couple of weeks with nothing in it and plant it, and didn't have that reoccurrence. Again, we didn't observe any whitefly. I'm not saying they weren't there or there wasn't some other insect vector that could have also potentially done it, but it wasn't observed. So
yeah, I mean, these sorts of reports are likely to come up, and now, embarrassingly, I actually forgot that we had this conversation in the moment, but that's true. And that's one of the reasons why I really like to get insight from Stephen here, because that was a really great conversation that we had about that previously. But yeah, LCV interactions are very likely to continue to propagate as cannabis kind of continues to languish in that regard. Many people are just unaware of it, and the way that people share clones and other sorts of tissue is going to have that effect. So the Spodoptera are the armyworms. They are a genus of moths, They're probably not the worst genus of moths to get into cannabis, but they are one that I feel like people don't hear about a lot. So I wanted to focus on them a bit here. Other moths that people are dealing with more so are probably the tobacco budworm moth and the European corn borer and the corn earworm, all of which are very well known for boring into the flower bud, and then they defecate in the flower bud, and then the flower bud becomes moldy. Or also the wounds could transfer the various pathogens like botrytis and other sorts of fungi that just ruin the product at the end. These armyworms do not do that to that degree. They are not really typically flower borers or even stem borers like the hemp stem borer. But they do constitute a problem because they are great defoliators, and like all of the Lepidoptera, the thing that they do really well is they grow rapidly and they eat a lot and they continuously eat. In fact, caterpillars, if they don't get enough food, will have major problems because they're literally built to constantly eat unless they have some specific species adaptation that allows them to not do that. Most caterpillars have a very quick life cycle of a couple of weeks, but some caterpillars take like multiple years to come to adulthood, believe it or not. But you won't be dealing with those with cannabis. Here I have two Spodoptera, Spodoptera praefica, which is the western yellow-striped armyworm, and Spodoptera frugiperta, which is the fall armyworm. And they're both pretty common in North America, especially in the west of North America. And this is about the season you'd probably be seeing them. In fact, I took this picture on the left 6 days ago. So you can see that, and also for an example, you can see what the frass of a caterpillar of this size constitutes. And this was after I let this specimen feed on about 2 like really, really small dandelion leaves. And this was after it actually, I was doing a test because it had actually vacated its entire digestive tract already before I gave it the food. So I wanted to test how much volume you got going in and coming out. And that's actually quite a bit. So even a very, very little bit of feeding can cause a large amount of frass to come out the other end and ruin your flower. Yeah, and they developed about 18 million years ago, and they tend to feed on your legumes and your beans and that sort of thing, the Fabaceae, your peppers and tomatoes and tobacco and that sort of thing, your Solanaceae, and also the mallows, which are in the Malvaceae. So that's just sort of an example of what they feed on. They're generalists, they'll feed on woody plants and herbaceous grasses as well. And here we even have an example of some of their adaptations. There's a diagram from a research report, which I didn't cite, unfortunately, dang. But if you want the citation, I'm happy to give you the research report. Yeah, so you can see how they've evolved over time, and you have 2 major types. A type that specializes on grasses because of their sort of tough silicate leaves, and also those on woody herbaceous plants, dicots and monocots, essentially respectively. And that's it. For this presentation, I actually wanted to give a pretty good amount of time for Q&A. But again, my name is Mathew Gates. Zenthanol Consulting is how I operate. You can find me at SyncAngel on Instagram and Twitter, and also at Zenthanol, which is the same channel that I will be interacting with in the chat. Yeah, so hope that was illuminating. And if you have any more questions, if you want me to share any research, or if you want to check out some of the research on my YouTube channel, please feel free.
All right, we got a ton of questions. That was a really awesome presentation. I guess first off, can you please go over Septoria a little bit more? I've been getting crushed by it here in Michigan a few years now. I've sprayed just about everything except fungicides like copper sulfate. Do you want to touch on that, and then I can touch on what I found works here in Oklahoma?
Yeah, absolutely. I found that a simple suffocant like Sulfoyl X was pretty useful as well. I'm sad to hear that this person's having so much difficulty with it because it can be kind of pernicious, and I do have trouble with controlling it if it's already kind of become a huge problem. That's why crop scouting is such an issue or such an important aspect. If it's a very small infection, then you can kind of control it really quickly with a spray. But I feel like it becomes more and more arduous as it multiplies in the crop.
Awesome. Sorry about that. A little bit of a delay there. All right. What other questions? Oh, so what I found for Septoria is when I first see that first sign of it, I just hit it with a Bacillus pumilus, then a Bacillus subtilis, and then a good lactobacillus, kind of on a rotation every 2 or 3 days, hitting them with one after another, and then coming in with a good liquid IMO to kind of give it the competitors and a new fresh inoculation. That particular procedure, you repeat that twice, and it seems to, hit it pretty much out of the park here in Oklahoma. You do have to catch it early. Once it's progressed to a certain point, those plants pretty much are gone, and you can't really reverse it once it's past, progressed beyond more than 25 to 30% of the plant. It can be really hard to reverse after that.
Yeah, don't forget in some of those situations too to not just replace your plants, but a lot of times replace the soil that you're working with, or at least treat it. Too many times I've seen people will go through and clean their entire grow and swap everything out and then reuse the same likely infected soil. So, just remember to also consider that in your cleaning process. If you are going to pull something out or start over, if something has gone too far, don't cheap out on the soil. Make sure you at least treat it if you're going to reuse it.
I definitely agree with those two things, especially the biopesticide of Bacillus subtilis.
Yeah, those are really good. The best one I've found out there currently is a product called Cease. I don't know if you have a different one that you're a fan of.
I like Cease myself.
Yeah, I found the best combo in general for biocontrols for mold is alternating Cease and Sonata, which is the Bacillus pumilus. Seems to work really well. I'm trying to find the other questions. There was like 5 questions, and now I can't find them here. Okay, maple trees have Septoria. It doesn't seem to bother the trees, but I've wondered, should I spray the trees to protect my cannabis?
I would say absolutely. That's definitely part of your sort of biosecurity assessment. Like I was saying for rice root aphid and other pests, it's very possible that plants on your property or plants that are just outside of your property, almost more annoyingly, can cause problems for you by harboring pests and pathogens.
Alrighty. Another question, what beneficials are safe to use in flour?
Oh, say that again.
What beneficial insects are safe to use in flour?
Yeah, sorry, I got a little bit of a garbled message, but it's a good question. I like to use, so when you apply biocontrol agents, some biocontrol agents, depending on from whom you're getting them, and what kind of product you're getting, they might have like filler. And so I just feel like it's very important that people are aware that they shouldn't just dump that onto their flower material. A lot of people kind of intuitively get this, but I just want to say it just out of the gate. Sometimes that filler is vermiculite, sometimes that filler is bran, sometimes that filler is buckwheat, and sometimes the filler also has a bunch of frass from the organisms living in the filler, through their shipment process. Biocontrol agents like predatory mites, like Amblysia swirsky, Neosilius cucumeris, Phytocillius persimilis, californicus, or Neosilius californicus. Your parasitoid wasps are generally going to be fine, and your predatory flies will be too. Lacewing larvae, that sort of a thing. I generally find that most biocontrols are fine to use in flower. The ones that I am reticent to use are typically ones that are part of a biopesticidal sort of application process. Those are the things that, because I'm not always sure, and every location is different. about what the inert ingredients are, whether that's going to have a problem. Is there an enhancer they don't mention, like piperonyl butoxide and pyrethrin products, for example? That kind of a thing. It just sort of scares me from a regulatory and compliance perspective.
Oh, what a great answer. We have another question is, have you had any experience with a product called Nukem? And does it work?
Yes, and yes, that's been my experience, but I don't remember the exact ingredients off the top of my head. Do you, Steven?
It's an
I don't.
enzyme-based spray, so it's yeast to produce the enzyme and a little bit of citric acid, and I want to say like potassium silicate or something like that, or some other agent.
Potassium sorbate. I think it's similar to Dr. Zymes for the most.
Similar to Dr. Zymes.
Yeah, I definitely remember the name, but I didn't remember the active ingredient. And I like to make sure that that gets said so that people who are thinking about using it kind of already know that and what they're looking for. They might have a similar product for a lower price point or a different formulation. Actually, on that note, I just want to say that pesticide formulation matters a lot, both in its efficacy, but also just like when you're considering, if you see two products and the active ingredient's the same and the amount is the same. One of the things I learned early for me in agriculture is that that is not necessarily the same product, and how it's formulated and the physical properties of it and any sort of like minutia in that process that you're not privy to could have a totally different effect in crop, both a positive one or a negative one. So it's just really important that you don't think that all the different potassium bicarbonate products are the same or something like this, or that all enzymatic products are going to act exactly the same.
I think a great example of this is Bavaria bassania. Each of those strains, Botanigard, that strain seems to infect insects at a different rate than Bioceres in different species. Bioceres seems to work better than Botanigard and vice versa, depending on the exact target insect. So, even incorporating the same species but different strains as part of your rotation can make a big difference, and especially prevention.
That's absolutely true. Just as a fun fact, in Botanigard, there's a Beauveria bassiana isolate GHA. And I've found that the GHA actually stands for grasshopper active. And the first Beauveria bassiana, if I remember correctly, I think I was reading some patents on it. And I think that it was first isolated from a cucumber beetle, Diabrotica diabrotica. And but they were trying to find a pathogen for grasshoppers and that sort of a thing. And I'm not saying that it doesn't work on other things. That's not why I bring it up. I just bring it up because like it's a neat holdover and it's a fun fact, I guess. But you're right. There are definitely isolates that affect various other pest targets. And I'm all for like a more targeted isolate because from an environmental standpoint, an ecological standpoint, my major caveat to Beauveria bassiana, which I love to use, is that it's a broad spectrum biocontrol agent in a manner of speaking. So being able to curtail that broad spectrum nature from an environmentalist perspective to like keep it from affecting things you don't want it to is something I advocate for.
Do you want to touch on successful grasshopper stuff? I have some cool stuff I was working on in Africa, but I know that there's Nolo bait as a popular one that people use for a biocontrol for grasshoppers. What have you found that works well for grasshoppers and the like?
Actually, to be honest, Beauveria bassiana is one of the ones that I found to have the most effect for me personally, and also the thing that you just mentioned as well. Not to like, I don't have a lot of experience with treating grasshoppers in particular. I tend to like to sort of advocate for and utilize physical barriers, just like at a greater degree. But those two intimate pathogens are really nice to use in combination with the physical controls. I find that combination works pretty effectively for a lot of people if it's implemented early enough.
All right. Someone else asked, do you agree that plants with high Brix levels are more pest resistant?
They can be, but not necessarily just because of the Brix. Because the Brix is the dissolved sugar content. And that will inform you of a lot of things about the physiology of a plant, like, for example, how much sugar is in it, but also maybe its photosynthetic rate. And so if a plant is at a higher photosynthetic rate, then you can sort of make the sort of assumption that ostensibly that plant is probably sort of physiologically doing good, or at least it's pumping out a lot of the food that it's trying to get from photosynthesis. But then, whether or not that translates to conferring resistance, because like I said, plants and various plants have different ways of combating pests and pathogens in different ways. And so how that happens, maybe that pathogen still has a great way to exploit susceptibility genes inherent in the genome of the plant. The Brix levels will have nothing to do with this. It won't really matter if the plant has a crippling exploitation that the pathogen can use, or a pest for that matter that's got some sort of behavioral or functional physiological way of mediating that. But generally speaking, a plant that's producing a lot of sugars is probably on its way to doing well for what it is, if that makes sense.
Absolutely. I think the other thing too is, if a plant's got a high Brix level, it's rocking, it's healthy, its immune system is at peak, peak production and peak ability to produce those extra terpenes and flavonoids and other things that the plant uses to defend itself. So, I think that's also the reason why when people think of, that's probably more the reason of what's actually happening with those high Brix levels. It's not that the Brix itself is doing it. It's an indicator that the plant is just happy and as can be and is capable of defending itself.
Yeah, I think that's a great way to interpret that. I have heard some people say that the sugars will be too much for the insect, and that the insect won't be able to feed on the plant because of the high sugar content. And from my own experience and also from the research that I'm familiar with, the literature on subject, insects adapted, or the herbivorous ones anyways, they adapted to an herbivore lifestyle. Carbohydrates from plants in the form of particularly sucrose is incredibly important. Aphids and other hemiptera can't get enough of the stuff. And when insects do break down those sugars in their bodies, they convert it to trehalose, which is a much more inert sugar, and it keeps the osmotic pressure down. So the insect can have tons of this sugar sort of moving around in its open hemolymph, sort of a circulatory system, unlike ours, which has vessels. And those sort of sugars, the trehalose, can be rapidly utilized by the plant's physiology, particularly when it comes to flight, which this is one reason why a lot of flying insects like to feed on sugary substances, because it's very energetic, right? It's a great resource for that.
Absolutely. One thing I was going to touch on that we kind of got a little bit sidetracked on. I had real good luck with grasshoppers, where we were collecting them in Africa and collecting a bunch of the grasshoppers and putting them into a pile and then mixing them with insect frass and then mixing them with rice and doing an IMO collection, indigenous microorganism collection. And then utilizing that into an IMO2, and then turning that into a liquid IMO to inoculate the plants with naturally occurring insects that feed on that chitinase and on those insects locally. So that can be a great way to target your local insects, collect them and mix them with some extra insect frass, throw them in your IMO collection, and actually go out and try to collect and target those. It can be a great way to start on your own journey in that direction if you're looking to do those types of biocontrols. Alrighty, well, Looks like we're about to transition here. Do you have anything else you wanted to mention, Matt, before we wrap up?
No, not really. Just to say that as sort of a community service, I really would appreciate if people are able to use me as a resource. So essentially, if you have identification issues, if you find that there's a pest you don't know, or that you need some help with something, please feel free to reach out to me. It's very important to me that I'm able to utilize this information. It fulfills me. It helps achieve an objective for biosecurity. Essentially, I look at my work as having this sort of prophylactic effect for as many people as possible. It does, and it all helps us all, the better we know about IPM in general. So don't hesitate to reach out if you are having issues, especially in this time where supply lines are difficult to ascertain. Situations in various parts of North America and other parts of the world are in great stress, both environmentally and biologically. So it's just very important to me to help as many people as possible within that context.
Awesome. Yeah, and be sure if you do send him stuff, be sure you send the location as much as you can. It helps him ID the insects as well. Alrighty. Well, much appreciated, and we look forward to talking to you again soon. Do you want to mention your podcast too before you sign off?
That is a pretty good idea. So so in regards to like questions and things, one on my YouTube channel, Zenthanol, I have an IPM FAQ series I do every month. So you can send me questions on my Twitter and social media and Instagram and also on YouTube. And I'll collect all those questions and I'll answer them in the video series. I also have the Cheap Home Grow podcast, which Spartan Grown is on here as well, and various other people. I do that every Sunday on the Cheap Home Grow, growing with my fellow growers. And then I've also recently been on a podcast with Eagle Gardens, who is also in the chat right now, episode 181, and we talked about IPM and some of my background. So if you're curious about that, you can check me out there too.
Wonderful. Alrighty. Well, thanks again for joining us. Appreciate it.